Hey guys, so in this video we're going to be looking at the structure of an antibody. So this is a structure of an antibody, a typical structure. So that means that antibodies come in different shapes. So there are 5 basic types of antibodies. We have antibody G, antibody A, antibody M, antibody E, and antibody D. These are the 5 types of antibody we have. However, this is just a typical structure of an antibody. Looking at the structure of an antibody, it looks complex, but we're going to break it down as simple as possible. The first thing I would want you to know about antibodies is that it has two chains. Two chains. We have the heavy chains and the light chain. The part in blue forms the heavy chain. So this part, all these parts here, forms the heavy chain. Meanwhile, the part in red make up the light chain. So we have this part as the light chain. After knowing that, the next thing you should know is the divisions of the antibody. The antibody is divided into 2 parts. We have the upper region and the lower region. The upper region is called FAB, F-A-B, and it stands for fragment of antigen binding. Fragment of antigen binding. Now we're going to talk about that as we move forward. The lower region is called fragment of crystallizable. Fragment of crystallizable. So FC. So we have the FAB region or the FAB region and the FC region. Those are the 2 major divisions. The next thing you should know about the antibody is that these molecules are actually linked together by what you call the disulfide bond or disulfide bridge. So all these black links are called disulfide bond, or you can call it bridge. So these molecules are linked together by disulfide bridge. Before I forget, the most important part of an antibody, although every part is important, but one of the most important part of an antibody is what I want to talk about now. You see, the antibody can be divided into the constant region and the variable region. The constant region and the variable region. So where is the constant region? The constant region are the region from this part down to this part. So you have for the heavy chains, you have C1, C2, C3. That means the heavy chain has 3 constant regions. Meanwhile, the light chain has only 1 constant region, which is this. So you have the constant region for the light chain, the constant region for the heavy chain. And the same thing applies here, C1, C2, and C3. These are the constant region. What's the function of the constant region? This is where most of the biological activity happens. So most of the biological activities happen in the constant region. Now the next region are the region on top, and this region is very important. They are called the variable region. So this region on top is called the variable region. And I will tell you why it's important. You see, this region, like the name implies, can change. It can undergo what we call class switching. So this region can actually change because it is specific in nature. It can change to fit in any type of antigen that comes. So basically, antigens will always bind to these parts, like the name implies, fragment of antigen binding. This variable part is the part where antigens bind. So when foreign body gets into the body, the antibodies quickly class switch their variable region to make them suitable to bind to an antigen. So the basic difference between the constant region and the variable region is that the constant region remains constant. It doesn't change. And most of the biological activity happens here. As a matter of fact, the constant region is actually fixed to the cells of the body or the cell in general. Meanwhile, the variable region is what changes. It carries out class switching. It can change. For example, if we have E. coli enter the body, it can class switch to bind E. coli. If we have another type of antigen enter the body, it can also class switch to bind that particular antigen. So it is specific in nature because it has the ability to change, to class switch. That's why it is called the variable region. So this is a quick structure, a quick run through the structure of the antibody.